Okay, let's get it started. So today we are going to discuss dimensionality reduction. It's also called dimension reduction. This will be the most important lecture during the whole semester. If I put a start somewhere on the different slides, but for this, the whole lecture will be very important in terms of this semester. We will talk about the three dimensionality reduction technique. PCA definitely in the center of machine learning. If you don't know anything, but you learn PCA, understand how it works, you probably learned 1/3 of the whole machine learning field. That's what I'm talking about. I'm not talking about the deep learning or other, but for this fundamental machine learning algorithms, you probably have already had idea about 1/3 of them. T-SNE and FIT are those newly developed, much later developed technique, and people widely use them for non-linear data points. And when the data points contains nonlinear relationship, we use them widely in single-cell data analysis because single-cell data shows a lot of those nonlinear properties. So we do need those new dimensionality reduction technique in order to understand the structure between data points. I will show you how. Okay, so before that, let's get a bigger picture why we are doing this. So through the lecture, we know we have using single-cell technology and droplet-based. There are other methods, of course, to get the single-cell rates and mapped into reference genome. And using cell ranger, we get this expression matrix or UMI count matrix. This is a gene by cell or cell by gene. Anyway, it will contain thousands of genes expression profile in thousands of cells normally per sample. And the next step will be we treat individual cells as a data point, as your sample. So you have individual samples. Each sample is a cell, and then the gene expression profile that's from these thousands of genes define the property of each cell. So we want to reduce the dimension. The reason is each sample, each cell, defined by thousands of genes, but those genes, you can summarize those genes' properties and use 3 or 4 or maybe 20 dimensions rather than thousands of dimensions to represent the relationship between cells. So that's why we reduce the dimension to 3 or 2Ds in order to visualize the relationship between cells. So that's the first step you see on this workflow or pipe chart, this typical workflow. And the first step is to convert this raw data, okay, the matrix, thousands of dimensions into 2D picture. So this is using T-SNE, T-S-N-E. So after that, so we will follow the workflow and throughout the different lectures. For example, next week we will discuss, once you have those distribution of the cells in 2D or 3D, then you can apply clustering algorithm to group them, depends on distance between them, and then make them different clusters. Then you can then look at different gene or feature genes expression in regarding to their distribution or abundancy in different clusters. So here are some examples of marker genes for each different clusters there. And you can either present in this heat map way or using violin plot to show relative expression level for those marker genes. So using those marker genes, try to figure out the cell type. This is the biology knowledge will be plugged in here. Okay, so that's what we are going to do, like a very basic pipeline from a raw data to the annotation of cell types. We are not doing any comparison yet. This is just one sample. We try to understand within this sample what cell type are in there and how many different cell types regarding the numbers of cells in different cell types. So that's what we're trying to do. Once you build up this pipeline on your mind and you know what to do, and then we can run another sample, and then at that stage we have two samples to compare. Okay, so the essential part of this is to do the dimensionality reduction. Okay, for visualization purpose, to enhance the signals, because there are a lot of genes which not very informative, so you still keep that in your downstream analysis will not very helpful, and also slow down your pipeline altogether. So at least two purposes. We want to clean the data and extract the major signal out of it, okay, resynthesize it, and also reduce the dimension so we can visualize the majority of these distributions, okay? This is not the whole picture. It's just from one particular angle, just like you look at the object and not look at the object itself, but look at the shadow of the data. But this is just one angle to see the data. Okay, so this is UMI matrix. So we already touched based on the high dimensionality, but I just formally go through this. What is the high dimension data? So each gene here in this table defined by those each cell, okay, each cell, each column, cell 1, column 1, defined by those genes expression. So you have to count how many genes. So if you only use the first gene to separate the cells, then you only have one dimension of properties. If you use top 2 genes, okay, then you have a 2D data set. If you use top 3 genes, you have a 3D. So the first gene will be 0, 8, 15. Second cell will be 0, 2, 1. So you can plot them in a 3D coordinate system. But once you jump to once you include more genes, you won't be able to directly plot them in computer in 3D. So you have to think about the 4D, and once you add more genes in your data set to describe your cell, so you have to imagine the higher dimension. Okay, it's a 4 or even higher, right? So that's why if I gave you the high dimensionality data, you want to summarize and make it reduced to the level that you can either plot the signal easily, and if you use 3D or if you use 2D, you can use multiple 2D and to represent combinations of a dimensionality to show, and you can to ease your downstream visual exploration of your data. So this technique, just you have a 3D object, then you crush that into 2D, okay? The same data points are still there, but you just visualize them different ways, then you get a slightly different impression of your data. So this is a t-SNE plot that we normally will see look like this. But before that, we are going to talk about PCA. I have a question for you. What your understanding of PCA so far, maybe from this side, and any answer? Have you used the PCA before, or what you have been told what a PCA is, from which side? Who want to mention PCA? Rashranos? Yeah.
We want to ensure that when we are having so high dimensional data,
Yeah.
reduce it to be structured, we can understand the data
Mm-hmm.
in a very less number of dimensions.
Yeah, so what you say is PCA is a dimensionality reduction technique. Okay, who else? No. So what you said is it's either correct or not, because PCA, that is a technique that you can use it to do dimensionality reduction, but itself, the operation itself will not give you the reduced dimension. So you didn't change anything. You just rotate your data points. And after rotation, if you want to do the dimensionality, that's you can do, but PCA itself is not dimensionality reduction technique. Okay. So what is the principal component? So who can explain the principal component?
The dot product is 0?
It's not a dot product. Yeah. So component is, so you separate an object into x, y axis. So component basically means the coordinate system x, y axis. Okay? So you have x component and y component. If you reduce to 3D, then you have a z component. So component here is the the direction of your coordinate system and the x, y, z, those axes. So principal means the major. Okay? So you want to find the dominant component. You want to find a new axis for your data. Dominant, which to show the most variable within your data. Okay, so this is called the principal component. So this analysis is try to find a rotation matrix to rotate your data a little bit, okay, to help you to use the principal, the first and second and third axis to view your data. The second method we will discuss will be T-distributed stochastic neighborhood embedding, okay, TSNE. Yeah.
Isn't PCA, whenever you're determining which ones to rotate it by,
Mm-hmm.
don't you determine it by the largest eigenvalue?
It will. Eigenvalue is an approach you can get analytical solutions. It's a shortcut to compute. You don't have to use eigenvalue. You can try different axes. You can try millions of them, use optimization to find the axis. Yeah, but the eigenvalue will be an easy way to find it very quickly. Yeah. Yeah.
Can you say that PCA is basically choosing the most important variables that can explain our data?
Yeah. So it depends on how you define the most important.
Isn't it like the ones that can create an identity matrix?
It's not.
No.
No. Yeah.
Ones that explain the most
Yeah.
variability?
Yeah. Yeah, so if you look at this 2D example, this is 2D, then your data point, if if your data point like distributed like this, like this shape. If you are a 1D animal, so you will work around in this region. You will find from here to here is the longest range you can get. So you want to work around this axis in order to fill the whole magnitude or distribution of your data. But if you use existing coordinate system and you run between unrotated coordinate system, you just only fill this range, which not as broader as the data should be.
So you just rotate it to center the data?
That's right. So PCA will help you to find we should rotate. Instead of using this, we should use this as my new axis and use this my y. So this is my x, y parameter. So this original was green. Okay, original was green, and based on the green, this 1D animal, if I only live in x-axis here, I only fill my data points in this range, but not as broader as it can be supposed to be. So now the question is how you compute this variability. Okay, so this is, say, V variability, and this was original V. So how can I compute that? You can compute by pairwise distance between those points. You can see I compute the point 1, 2, 2, 2, 2, 3, 1, 2, 3. You can compute all the distance between them along the axis with their x coordinates. So how wide, and then you compute all the distance 1 ij basically, and then you take a square and then sum together. So basically, you just look at the difference between all the data points. You can do this variability for along the x-axis, and then you do this along y-axis. So you try to learn based on, for example, my current green axis system, green coordinate system, how along the x-axis, how my data points are differ from each other. Yeah, so if you use, for example, this one, you use one gene for this one. I translate that into single cell data. Okay, so this is an AHR gene. Okay, so in your y-axis, you use FS3, so two different genes to define your cells. And then you compare first. For this particular gene, my cell 1 different from cell 2, you can just 0 minus 1 maybe, or 0 minus 3. So you compare all the difference. And then you summarize them together, basically. That's your variability along this gene. Then you compute a variability along that gene and to fill how widely difference between your all the data points. Okay. But we just hypothesize that those cells are the black dots here. If you don't rotate them, and your variability between cells for this particular gene, the first x-axis will be anything between them. So you find the difference and take a sum, okay? Square them because the distance may be negative, so you take a square, it will be positive. And then you project your data to here as well. So this is before rotation. Project the data to y-axis. For this particular gene, you look at all the cells difference. Then you take square and sum them together. So these two values give you the x-axis and the y-axis variability for the distribution of those data points. Is that okay? Yeah. Then I can rotate my coordinate system the same as rotate the data. Okay, I rotate this. I use this blue system as my new system, and I can do that again to look at the distance between those genes. Okay, the number has been rotated, and look at them, the difference between data points, and compute the total. Basically, you take them together, put them together to make this clear. So for the variability, you look at the difference between gene 1 and 2, square them, plus gene 1 and 3 squared plus gene 1. Then 2 and 3 pairwise, everything put together. This tells you how differ or how from each other for those data points. Okay? So our job is, how can you find this coordinate system? So from the green one, and how can you find these blue ones in order to maximize this disperse between data, okay? By the way, for this rotation, for this coordinate system, let's see. This system for the original one, the green one, was defined by A. So this will be 1, 0, 0, 1. Okay, two vectors. So the first vector, first column, is your x-axis, and y is your y-axis. So we want to find the blue one, so we have to find the alternative. We need the 4 numbers here, okay? We need A, B, C, D, 4 numbers. So these 4 numbers, the first 2 will define the vector from x-axis. So the first column, AB, will have a vector, okay? And the CD will another vector which goes to here. Okay, so this is your new axis, x-axis, that's the new y-axis. Is that okay? So we need 2 vectors to define this. And because they are orthogonal to each other, so we know the dot product between this and that has to be 0, okay? So it's easy to achieve that by put B here, A here, and put a minus sign here. So, so this matrix, although contains 4 elements, but it's half determined because we require the x and the y axis has to be orthogonal to each other. So if you write in this way, it will be okay, and they are orthogonal to each other by default. So that we only needed to optimize or estimate 2 values. Okay, so the last dimension, the last axis has been self-defined. So that's what we want to solve, to find a rotation matrix which all the axes are orthogonal to each other, and we can use this as a new coordinate system in order to rotate my data, okay, use the new coordinate system to show the maximized variability between data points. So that's PCA. So to do this, we need a lot of preparation before we can see, okay, why people compute the PCA in this way. So the first background knowledge we should have is this picture. And we know if you use B as your new coordinate system and you project A onto the B, and you can use this to compute the you can use this dot product to compute the projection, how long this part is. Okay, so A's coordinate was determined first by this x-axis, which is horizontal, and but it's a new coordinate if you use B as your new x-axis. So the coordinates for the A should be this one. This one is A, the length of this times cosine theta. The value is this. Okay, so the dot product between A and B. A is your original data point. B is your the vector. It's not this B, but in this case, the B will be the vector you used as your new x-axis. So you project this onto B. So the length of this is your new, under your new coordinate system. What's the x coordinate value for A original? So it's just A times B, so vector multiplication, and the normalized by B. Okay, so if you project this A onto 1, 0, nothing will change. You still use your original axis as your coordinate. So you can try, if you have a value, say, at the position 2.5, 2.5, then you multiplicate by the x-axis, you will still have 2.5, 2.5, because that's the position of this original. But if you use different x-axis, you basically will project your data onto the axis. You change the value. To compute that value, you just do the dot product. It will still be very hard to understand. We can use example data sets to play with, and you can figure it out. So another concept is this eigen value, eigen vector, this system. So we will use this concept very, very soon. Remember this. This A is a rotation matrix. So its coordinate says this A. If you use A to rotate a vector x, and for this particular special vector, then there you don't have to change the direction, but it only multiplicate by a constant. Okay. Basically, make this vector longer or shorter by a factor, constant factor. And this form is you want to remember, and this, we will use this later. So if you transform this, you can write A equal to, I don't know, should be this or that, but one of them will be the inverse of this matrix or vector. And you can rewrite this relationship in this. Or you can write lambda, so this is a constant, becomes transpose A. Okay? Basically, you put those variables or matrix or vectors, replace them to rewrite something like this. Okay, so we are going to use this equation later. Okay, not now, but because of this relationship, you can decompose vector matrix like this, or rewrite this into a constant. Okay. So I just reused some of the slides from a previous lecture. So this is just another example to show this is the matrix A, and then you have two eigenvalues, either -5 or 2, and each of them have a corresponding vectors. So the first vector is this, and another is this. So this one is normalized. This should be in the unit circle, and this one will be scaled. But they are the same. Okay. Okay, so PCA example. So this will be an example to reduce dimension from 3D to 2D. And we will play with this example in MATLAB, and then you have a cloud of data, and you see some of this access you expect to see, because there some points will help you to disperse that away. So once you run PCA, and then you use this first principle and second principle to see the data. Okay, you actually reduce the dimension from 3 to 2. If you don't do PCA, you can reduce dimension from any angle. You can see from here, and you'll see, okay, it's fine. You can from this angle, but the PCA is a way to give you the angle. Give you the angle of the view, okay, to maximize the distance between the data points. So that's what I said. PCA is not a dimensionality reduction technique. You can use that to find the rotation which maximize the separation of cells, the data points, once you want to reduce that dimension using first and second principle or third. But if you don't do PCA, you still can do dimensionality reduction, because you can see I have a 3D data, but I only look at them using X, Y, and forget about the Z. You'll still be able to do a dimensionality reduction. So this is a data point, the data set I this is actually a data set. Okay, I just add some noise to make this make this as an original random data, but in reality, this is the points on the T port, and then you rotate. As I said, PCA just gave you this view. It's not a coincidence because people want to see from that view to see the features of this T port most. Same here. This is a real T port, and I tried this. This website is gone, so you don't have that product on their website. We had a World Market in College Station, but they shut down during the pandemic. Okay, so PCA is based on lineage algebra, so it's not machine learning, not a deep learning, not a neural network. It reveals the hidden geometry of structure within your data, which from different angle maybe you cannot see. So if you take a picture from the top, you only see this feature, okay, the handle, because the handle becomes just a line in the shadow. You don't want to take a picture from this angle because nobody sees this. So to learn this, the theory a little bit challenging, okay? So all this, why, okay? How can you do this? But so try to make it, try to make you understand that when you compute it, it's very easy. So to do this, there are built-in function everywhere, all the languages you should have. Just run PCA and they give you the results. The result, what is the result of PCA, out of PCA? The result of PCA is this rotation matrix. It just tells you what your new coordinate system should be. It was 1, 0, 0, 1. This is x, y axis. But the new one will help you to, is a rotation, it's a new coordinate system, a new x axis, a new y axis. If you have a 3D data, then you coordinate system was 1, 0, 0, 0, 1, 0, 0, 0, 1. So that's your x, y, z, right? So this is your x, y, z. X, y, z. If you have a higher dimension, this will be just increased. Okay, this is 3D. You can have a 4D, will be a 4 by 4. The PCA will give you, if you inputted 2D, it will give you this 2 by 2 rotation matrix, allow you to use it as a new coordinate system. If you gave data 3D, so that's your original coordinate system, they will give you a 3 by 3 coordinate system matrix as a new system. And if you give 4D data, it will give you a matrix 4 by 4. That's a PCA will do. Okay, so PCA gave you this rotation matrix, okay, rotation matrix, to allow you to rotate your original data space, rotate the whole space. And the description of your data will change. So it was based on your original coordinate system. This value, for example, will be x1, y1, but once you change the rotation matrix, it changed, it will have different values. Okay, so how can you compute this? So I have data 2D. I will get the new coordinate system. I have 3D, like my T-port example, 3D data. I should have a 3D matrix, 3 by 3 matrix. How can you find those? If you don't know anything, you can use the built-in function. So like MATLAB have PCA. You give the data matrix and make sure you know columns are the variables, rows are samples, and then PCA function will give you the rotation matrix. You can also treat this as an optimization problem. And what that means is you try different x-axis. Because now you know for this 2D example, you have 1, 0 as your x-axis. You can change the angle. Okay, you try 360 degree the vector and try each of them. See which one gave you the maximize this dispersion of this value. Then you stop there. So you can achieve that. It's doable. But for high dimension data will be difficult because you have to find the first PC in the high dimension. So you have a lot of variable. In this case, you only have 2 values to rotate this. But for 3D rotation, you have to find 3 values as your new axis, x-axis. For high dimension, you have a lot of parameters to compute. So that's why in real computation, we do this. So this is the method we will use to first to get covariance matrix and then take eigenvector of covariance matrix as your PC, as your principal component. So what's the principal component? Principal components are the new coordinate system, x-axis, y-axis, if you have 3D z-axis. So eigenvectors of covariance matrix is the solution. We also have this way to use singular vector decomposition. So this is a matrix decomposition method. We are not discuss here, but may talk about this later when we have a decomposition lecture. So SVD is also another way to directly compute those principal components. And there are also so-called alternating minimization. So this use EM algorithm to do this iteration to find the solution. There are several others, and you can find the source code in this website here. So I will very quickly and show you the framework of the optimization, and we will have a lecture maybe 2 weeks later, and on optimization problem itself, 45 minutes. And Luis, can you stand up? And Luis will give us this talk because he's an expert on the optimization. Okay? Yeah, thank you, Luis. We will work out to see whether it will be after spring break or next week after spring break to do this optimization. So optimization is one of the most important topics because a lot of solutions, if you cannot find those elegant analytical solutions, you just optimize. You try different numbers, the combinations of numbers to give you accepted or minimize, maximize the solution or nearly maximize the solution. So it's a kind of a computational art, and how to configuration your system in order to find those combinations of parameters and solve your problem. Just like here, how can you find this A, B, or two numbers very quickly in this particular problem here? Okay, so built-in function in PCA, that's a documentation. You gave the X matrix is your data. Remember, you have to make sure the row and the column, which one is the sample, which one is the feature variable. Translate into single cell data is which row should be the gene, which whether it's a gene by cell or cell by gene, we have to figure it out. Otherwise, you build a PCA for genes, not for cells. So what we are trying to do is for cells, okay, to separate the cells. And if you ask for one output, this coefficient will be the matrix, will be this, okay? If you give 2D, this will be a 2x2 matrix. Rotation matrix is called coefficient as output. So this is the most important. All the other variables, if you ask, the PCA function will give to you, but you don't have to. This is the only one you need, because once you have this, you can transfer or rotate X and get a score. Okay, so this is like a derived values once you have this. And this latent is how much variable you explained for each of these PCs. They all can be computed secondarily. Okay, optimization. So in this case, in this cartoon, show you the optimization problem, and you assume your center is here. Okay. So we can use any. Let me stop this here somewhere. Yeah, so you can use any direction as your. So now your axis here, right? And then if you use this as axis, and then you project all your data points onto this new axis position, and then compute the distance between them along this, their separation, those project dots onto this axis. Then you compute their difference. And we can see if you rotate to a particular region, let's see this one. Is this a good solution? And then if you see the separation of the cell, these right dots are from the smallest here and largest here, but within between, they're not very separatable. They all class together, so pairwise difference between those right dots are not so high. Okay, this is not our best solution. So we try different angles. Okay, so this is even worse. Okay, the right dots even more close to each other. They cluster together, not a good solution. This is basically the worst, okay, almost. You see, when the blue, the original data points project onto this new axis, new x-axis, their axis values, they all very similar to each other together. This is not good. So our optimization procedure will try different rotation, and then each time I compute this total variable between data points. If it's too small, I will try another one until I found the solution So this is not good. Now I start to see the increase. And the best solution is here. Okay? So now it's the most separable position. I will stop this optimization and then stop there. Now if you compute the distance between those points, they reach the maximum value you can get. So this is the top three lines here is to use built-in function of PCA to compute the rotation matrix coefficient. Well, the rest of them are using optimization to find the same solution. This is a way to use a function called a function minute search. Okay, this is a built-in function for MATLAB to solve optimization problem, and you have to define a so-called penalty function. Okay, so your goal is to find the combinations of parameters to give you the minimum values of this objective function. So within this objective function, there are differences between data points. All right, so this is a total difference between data points. After you project your original data project onto your axis, okay, the new rotation matrix. Now I have to explain why. So as I said, we use eigenvectors of the covariance matrix as our solution, as our output of the PCA rotation matrix. Why you can achieve that goal? It sounds very complicated because you need to find new x-axis and have a y. If you have a high dimension, you have found all the values in this rotation matrix. But our solution is simple. We just get a covariance matrix and take eigen decomposition, and the eigenvectors are the PCs, are the principal components, are the rotation matrix values. Yeah?
Why would you not use the built-in function?
The built-in function is also calling this. So I try to explain what's going on inside the built-in function.
So we could use the built-in function.
Yeah, you don't have, you can understand this, but you don't have to, because everything, the individual steps have been synthesized inside this built-in function. Okay, so first we need to know how to compute covariance matrix. This also copied from previous slide. Okay, if you have two variables, you centralize your first variable by minus its mean. You centralize second variable by its mean, and then you take product of these two centralized data and sum them together. So that's your compute covariance between two variables. In this example, I didn't have spent much time last time, but we can talk about this. Now your input data is A. In this A, you want to compute the covariance variable matrix between three variables. So column 1, column 2, column 3. in this particular example is their student score in math, physics, and English. So variable name MPE, so MPE. So those are the 5 different students, their scores. So we want to compute the covariance matrix, not between 2 variables, not between math and physics or math between, we compute pairwise 3 of them together, okay? So you can do pairwise, you can do math against English, math against physics, and just use this method to compute the covariance between math and physics, between math and English. So you can get those values. But you can also use this matrix form to compute them all together, okay? So what's a matrix form? The first thing is you take average. So you know for first variable, average is 60, and the second variable is 50, and third variable is 70. And then each column minus their average, you centralize your data. Okay? So this looks messy, and we don't have to use this because try to write in the matrix multiplication way to do this. Essentially, you just minor every values here to its average. So you 90 minus 60, 90 minus 60 minus 60. Then you get this value, right? So jump from here to here, you just centralize your data. So this data is called small a. So big A, after minus each variable by its own mean, and you centralize the variable. So once you finish this step, to compute the covariance matrix, you have A transpose times A itself, and you do this transpose to itself, and that's covariance matrix. Okay? You can normalize by the numbers. You can divide by 1, 2, 3, 4, 5, or should be 5, because n minus 1. So you can divide all these values by 5 to get the covariance matrix. But that's the covariance matrix computed from the centralized data. So that's what I want you to remember. Okay, let's take a 5 minutes break, come back at 5 past 11. I will prepare some writing on the board here, okay? Okay, let's come back and let's see whether I can explain this clearly. So what do you see on the screen are this mathematical definition of a PCA using eigendecomposition method to compute to get this U. Basically, this U is what you want to get out of this PCA analysis. As I said, you want to get a rotation matrix. The rotation matrix is this U. So the first step, you have your input data big X by the dimension of n and p. So n is the number of samples, p is the number of the variables to define these samples. Okay, so in my data example, I have a matrix which is variable by data, so it's transposed. The same idea. The first step is to remove the mean, so you minus each element in the big X by that variable's mean. So in my case, I had a data example X, so it's here. So I have my example data, 2 variables, 2 rows, and 3 data points. And for each row, I compute a mean and use the value minus its mean, so I get this centralized data. So if you sum them up by rows, they should be 0 because data are centralized. So that's what the first step is. Okay, input data and remove the mean, center the data. Then you compute covariance matrix using this centralized data transposed and timed itself and divided by the number of samples. So this part is this corner here. You centralize your data and transpose the centralized matrix, multiply it by itself. And then here, the example doesn't divide by its mean, the number of samples, but this one might divided by 6 or n minus 1 will be the covariance matrix. So this part is this, okay? So you get covariance matrix C. Then we come back to eigendecomposition. In eigendecomposition, remember we write A as your matrix, then you have an eigenvector matrix. And when you multiply these two matrices, and the same as a constant times this matrix, okay? So I just changed the variable. A changed to C, X changed to mu. Lambda is still same, so same format. Okay, so this is eigendecomposition, and you rewrite that. You decompose the C matrix, which is a covariance matrix, into mu times lambda times inverse mu. So for any given covariance matrix is a symmetric matrix. You can get real values decomposition for their into these 3 parts. And these 3 parts, mu is a rotation matrix. That's your solution. Okay. So mathematical description is not hard. The computation just take a couple of steps. Negative the center, centralize your data, minus mean, and then decompose. Compute the covariance matrix and decompose that and get it. That's the steps of the computation. So I have to use a small example to show you why this procedure worked. So my small example contains 3 data points, V1, V2, and V3. And what I showed you here is my centralized data already, okay? So that's what the example is, okay? First, I will compute the covariance matrix from this V. Do you remember the formula? The covariance matrix is C is V transpose times V itself, and then divided by 3 samples. By the way, because my data matrix is transposed, so I have to write in a different way. Instead of transpose first, I have to transpose this later and divide by 3. So that's my covariance matrix in this form. So I compute in MATLAB using this and get the covariance matrix, okay? The value is the same. I use a built-in function, so I just validate. This is the same as that. So I just leave this like this for now. Now I'm going to compute, use my E as my new axis. So this is like my new axis vector. So I'm going to use E as my new axis vector. So this one was old. This old is 1, 0. The old one is 1, 0. And this is my new one. I pick, for example, this one as an example. Now I have to compute the after rotation. If I use this as my new axis, what's the axis x coordinate value for V1? So for this value, it was here. Okay, so this. Before rotation, it has a value, say, x1, y1. So this has a x2, y2 position. So those are the positions. But after you consider this as your new axis, you have to project this value. Okay, you project your vector, so that's your vector V1. You project the vector onto the new coordinate system. So your new x is here, so that's your new x1 new, okay? Remember this is a new x1 here, and your new V2 is project onto U2 here, okay? You project yourself onto this new axis Then for the V1 become here, okay? V2 become here, V3 become here. So those values indicated by this red vector are the coordinate for new V1, V2, V3, their axis value in the new coordinate system. So to write this is to project V onto E. Yeah, some transposition, okay. So this dot product is between centralized V times this vector. What it does is to project each V1, V2, V3 onto E to find its projected lengths. These are their distance from a new zero, okay? They're how to see? This value is the length of those new V1, new V3, new V2. So those are you can consider them as the distance. So the distance of D1, D3, D2, which is the distance between origin to the point which each data project onto this E axis. So you have three distance, and you multiply, square it, and take a sum. So this is a variation or variability. That's what we're doing here. So this basically equal to D1 plus D2, D3. In this numerical example, I also divide by 3. So basically this average so you still have to refer back to this dot product, and one of the application of dot product is to find the projection. We have to go back to here. So basically, that's the dot product A and B is to compute the way to compute the project, how long this projected distance from A projected to B. So here we change it to V. So three different V computed together. Each V you project onto E. So E is a B here, and those are V1, V2, V3 are the A. So you project your V onto the E, and the dot product is how long this projected distance is. So those distances are the D1, D2, D3. So you compute, you sum them together, is your variability of your projected original data in this new x-axis. What's the dispersion is. So this is the total sum of the variance, the difference between data points, basically the same. Okay. So you can imagine if I found a rotation, then V1, V2, V3 project onto this particular angle new axis, and their D1, D2, D3 are the largest, then that's the vector I want to find. So this gives you the variability after you project it to your new axis. Well, what we found is this value here, and that is 0.4138. So this value, okay, in our small example is 0.4138. This value is equal to E transpose, that's vector, multiplied by the covariance matrix, multiplied by E again. So this number is the same as this one here, okay? This seems unrelated because the covariance matrix is between three variables, two variables in our case. They are correlation, basically. But if you do this multiplication to your new axis vector, it gave you a value which is same as a projected distance, the summation. So by chance, it's not by chance, but internally they're linked. So in order to, our goal is to find an E to make this number as big as possible, right? We want to make this, find a new vector E to make this value the largest. Largest means you separate your cells most. So the same as we try to find an E to make this expression the largest, because they are same number. So that's why we translate our question, our required solution, rather than maximize this value, search for different E rotated. We just compute to make this large. So we translate this total variation problem to maximize we translate this into to maximize this. Maximize an expression E times C. Again, E is our rotation matrix, is a new axis. We try to find the largest number of this, because once you found the largest number you can achieve using a given E, this value is the same as you maximize the total variance of the projected points onto E. So if you look at this form, it's the same as, if you remember, I read this eigen decomposition, C mu lambda mu. So C is mu lambda mu transpose. You can also, from this, derive an equation. Maybe lambda is mu transpose times C times mu. So you see the similarity between this equation with that one. So to decompose the covariance matrix will give you this eigenvectors, which if you found the largest of the eigenvector, that's the solution for this equation here. And their corresponding eigenvalue, eigenvector is the E we searched for. So at the end, you gave this particular optimization problem, gave to the eigendecomposition equation, basically. It's definitely a concept jump a little bit, and but if you know those slides with the star on it very well, you see, okay, every operation here makes sense because this is the projected distance, and the summation of the projected distance are the transformed data points onto this new axis. So this value is how disperse of those transformed data on the new axis system. And this is total variance. You want to maximize this, and its value is the same as this one here. So that's why the eigen decomposition problem jumps in, because the format of this you can get from the largest eigenvalues. Yeah, so just give it a try to explain this a little bit. Try your best. You don't have to. This is the same as what I try to draw on here. So on your left is you use you're not using covariance matrix to compute those distance, project distance, this is total variance. On that side, if you decompose the C, and it's also, this equation is also gave you the total variance, the same value here. Okay. If you need more, and this YouTube video give you more explanation, yeah, you can give it a try, see whether you can follow that. In the middle somewhere, you need this Lagrangian multiplier, right? So in that case, we need Lewis to give some idea why, so how this linked with the derivative here. So I give you my simple explanation using eigendecomposition, the formula here, okay? All right, there are some other mathematical details to guarantee the eigenvectors produced out of this procedure will be their rotation matrix directly. The rotation matrix has to be all the vectors within the rotation matrix has to be orthogonal to each other, and also has to be normalized. So their mathematical guarantee that out of this, the rotation matrix mu is normalized and also orthogonal to each other. So it's mutually orthogonal. Okay. This is because this C matrix is symmetric. Covariance matrix is symmetric. So this is mathematical proof that you decompose a symmetric matrix, you will get this eigenvectors when they are orthogonal to each other. You have to this x, y, the dot matrix has to be 0. There are limitations of PCA when you apply to more complex data, and it's for example, this particular case, you have a perfect sphere of the cloud, then your PC will be randomly pointed to any direction because there's no direction give you the largest variable. They're all the same. And in this case, you found this axis, but you miss the other structure. In this case, you have this oscillation of this sine curve. You don't see those changes. You only found major components. In the teams, where's my team? I also have an example. This is from a PNS paper. For all this data, and you produce the same PC. First and second components are the same, although you have a very different shape of data points. So PCA will not be able to tell the difference using first two PCs to separate those different shapes of the high structure within the data. That's why we do not use PCA directly as our final results for the single cell data. So this is like a single cell data. Use the PCA, you get something like this. But there's some real structure within the cell population. So you'll only be able to separate them use just first two PCs. Okay, as I said, the data points was still there. You only rotate your coordinate system. You only rotate your view of the points, but still data cannot be separated by 2 or 3D. That's why we don't use the PCA directly in single cell data analysis, but we use PCA as a feature extraction or feature enhancing procedure. So you run PCA, but not using first 3 PC, but use first 200 PCs, and to replace your original data. Your original data is defined by, for example, 6,000 genes, but now you use the PCA to learn the first 200 major PCs. Now the dimension of your original 6,000 dimensionality data becomes 200. So this data set is much smaller than before, but captured the majority of this variability among the data. So we normally use this dimension-reduced PCA processed data for the downstream. So even for the t-SNE, okay, you use the PCA output as your input for next step. So what's a PC, what's the t-SNE different from PCA? As I said, PCA will not change your data points position. It's still there. It's still, you just rotate everything and at the same time. But t-SNE are different. Okay, t-SNE will change the position of your cells in a low dimensional space. Just like this, that's like underground map. If you don't transform your data, the physical distance between different stations may be very crowded. Okay, several blocks, you have another stop. And you have to distort the data to see them. And in order to draw this visually appealing or visually separatable the data points to somehow enlarge those crowded station distance longer in order to write the name of the station at least. So t-SNE are doing something similar, but still we have to guarantee this stop, the next stop should be the one without the transformation, without the distortion of the data, right? You cannot change the topology. You change the distance between data points, but still keep the relationship between their relative position. Okay, so that's what t-SNE will do. t-SNE will learn a probability first. So the t-SNE result, the earliest idea was from Jeffrey Hinton, and this is which year? 2003. So his paper is about stochastic neighborhood embedding. When you talk about stochastic, and also the random number generator will be getting involved, remember. And this will, he defined a probability which between two data points, xi and xj. He will first compute their distance between these two data points. All right? And then he will divide this distance with the summation of all the data points distance among your data. Say if you have 100 data points and you found two of them, xi and xj, and you found the distance, dij. And then you divide by all the pairwise distance between your data. You have 100 data, so you have D12, D13, D14, and D23, and you sum them together. So this ratio is a P, okay? The P between I and J. So this ratio tells you whether this I and J, these 2 points, are the friends or not, okay? They are neighbors. What's the probability they are neighbors? So in this example, we are having this 3D. Then in this 3D, there are 3 data points. The distance between cat and kitten is red and green distance small compared to dog. Okay. So the red and green dots, the likelihood between them neighbors will be larger than cat and dog. You can compute a p-value for that. So this stochastic embedding is, we will put all the data points from high dimension. So in this example is a 3D. 3D to 2D is not too hard. But imagine it's 6,000 dimensions. So you have data points. You know, okay, these 2 data points are more likely to be neighbors in that space. So we are going to regenerate. Remember the random number generation? We regenerate the same data point, but in 2D, and still guarantee the distance between these two points have the same probability to be neighbors. The likelihood are the same as there was in the high dimension. Okay, we completely reposite our data points in the 2D in a map. The only thing we will keep is whether these two data points are neighbors. The likelihood to be neighbors are the same as their original position in the high dimension. That's the only thing we want to keep. Everything else changed. I can put my 2 points here as long as they are relative to other data points have that property. I can put it here. So that's why in t-SNE, each time you run, if you use different random numbers, you get a slightly different picture. Sometimes your cluster was here, but in another run it was downside. But the relative position and their proportion values to be a neighbors will be still the same as they're in the high dimensional space. So we rely on some distance computation. You know how to compute the distance, and we just try to keep the being neighbor likelihood are the same. Okay, so this is just only 2 pairs of the two data points, 1 pair. So you have to guarantee all the pairs between each other. This likelihood are the same, map from high dimension to low dimension. So in order to compute this multiple likelihood probabilities, we use KL divergence, okay, to compute. To make a KL divergence as small as possible, we got to minimize this difference. So if you have 3 data points, they are likely to be neighbors in the high dimension. You have to guarantee they are likely to be neighbors in the embedded. So you just guarantee the P and the Q values overall are same, or as small as possible. That you match the relationship between data points. Okay. So you see, this is a completely different ways to compute the embedding. So the improvement to add in is by Mitten, and he's maybe he's a student of the Hayden, but they use T distribution to regenerate the 2D embedding in low dimensional space. Instead of using this normal distribution, remember I showed you the T distribution tend to put cells away from each other if they are too close to each other. So if you regenerate your neighborhood using T distribution, they are slightly dispersed a little bit to allow you to see the separation between subtle difference between cell subpopulations. Yeah, so they use Gaussian in the high dimensional, but when you project into 2D or 3D low dimension, you use T distributions to re-put the cells populations relationship. That's why you get this more crowded, getting crowded to solve the problem. Okay. Okay, why you need that technology? So if you have some shape of this, if you project, use a PCA or whatever, without change the position of the cells, then you will end up with, for example, all these points will, when you project into 1D, they all will be crowded onto each other. So you have to unrope basically to re-embedding to allow these blue cells to be rearranged in a low dimensional space. Otherwise, if you directly project them onto X or Y axis, you either blue cells and orange cells will be on top of each other, or if you look at that direction, then the blue and the red will overlap onto each other. So you really have to rearrange them a little bit. You keep this projected the likelihood. Okay, so in this case, for example, the gray point, the likelihood to be neighbors of the blue ones is much higher than they being the neighbors of red and orange ones. So how can you compute that probability? You look at the distance between these gray dots to all the other dots. Okay, they are very small to blue points compared to red points. And then you come back to this curve here. And the distance, you use gray point as your center, and then you move away from it. If the small distance, you see, look at its distribution value, okay, this curve, normal distribution. The likelihood to be the normal neighbor will be much higher, okay? So this value is much higher on this curve compared to those points which far away from the center. So basically, this normal curve tells you the likelihood to be neighbors based on the distance from, okay? So you want to keep this likelihood values when you change the position from high D to low D. If you use different normal distribution curve, you will get slightly different scores for those being neighbor or not. So if you see, okay, I use this sharp peaked normal distribution, and you have to be very close to each other in order to be high likely to be a neighbor. But if you use this more flat normal distribution, and you can move a little bit away from that, will not change much as your y-axis being neighbors or not. Okay, so that gives you some so-called hyperparameters. You can you can think about that with whether you want to neighbor has to be really a neighbor, or you have more relaxed scoring system to allow them to be neighbors. So that's a difference between T distribution and Gaussian distribution. Gaussian is this blue curve, and the red curve is T distribution. So T distribution allow you to be away from each other. Okay, the distance can be slightly larger, and you'll still be neighbors. So if you have a distance 2.5, you still be likely to be a neighbor by 0.1 likelihood. But if you use Gaussian distribution, you almost zero to be a neighbor to each other. So once you are not a neighbor, you can rearrange those data points anywhere on the space. As long as there's an empty space, you can put it in there. But you have to be neighbor, then you have to be very close in low dimensional embedded space. Okay, so here is you have neighbors. If you just by yourself, there's no real neighbors, you can put yourself anywhere, and there's a slightly different solutions. again, we have to use optimization. So that's why t-SNE will be slow, because you have to randomly give it a try. Say I have a new data points was neighbor for several other cells. And when you embed it to low dimensional space, you have to try this, juggle around their new position. That's why it's much smaller, slower than PCA. PCA, based on covariance matrix, that's easy to compute. And eigen decomposition is not hard, so very quickly give you result. Well, here you have to consider the distribution between cells, so many between pairwise between them, and then you have to find the solution which you stopped somewhere and give the output. Okay, I will leave a couple of slides to Thursday and talk about the fit embedding. All right. I gave the homework which allow you to explain explain PCA in your language. Try your best how it compute, and maybe you want to explain why you can compute that way. But if you can explain, for example, the procedure like this, okay, internally, and it's okay. All right.